So this, while we're interested in bone and muscle, this is going to become an increasingly important part of the national agenda. And recently, it was looked at in the New England Journal of Medicine, the cost of providing care for fractures and other diseases. And the 17 top conditions cost 50% of the Medicare costs for hospital care in the United States. That's 50% of the care of cost of medical care for Medicare patients. Number one is osteoarthritis, and this is hip and knee replacements. But if you look, number 3, number 8, and number 16 are also related to bone injury or fracture. Number 3 is fracture of the neck of the femur, hip fracture. Number 7 is complications of the device, and many of these are hip fractures that have not healed, and other devices not healed. And then in aggregate, other fractures account for number 16. And in total, 5 of the 17 top costs in the Medicare population are associated with musculoskeletal diseases. So this is going to be an important part of the national agenda. And non-union is one of the areas that remains a challenge. There are an intersection of patient characteristics with environmental factors and exposures and diseases that can account for increased risks of non-union. And clearly one of these is injury to the soft tissue envelope. And this is an individual that I took care of, was in a motor vehicle accident and had an open fracture. It was treated initially with an external fixator, and you can see an established non-union at this point. And this is the picture of the person's leg that after some reconstruction of the soft tissue envelope with free flaps. But this, again, went on to non-union, and this was treated operatively with a plate and screws. And the plate and screws, after 4 months, caused this to heal. And the question about this is what are the stem cell populations involved in this reparative process? Why did they arrest in this particular case? What are the signals that were activated simply by putting a plate and screw on and activating mechanical signals? And is there ways to deliver this more efficiently and effectively? And that's the purpose of bone healing models, is to try to understand these cells and signals and molecular events to develop therapeutic approaches. And in the context of that, the role of muscle, tissues, and cells as yet remains very poorly defined. The cell populations that are likely involved are listed, and the key part or initiating event in fracture healing is the proliferation, accumulation, and expansion of a stem cell population that subsequently undergo differentiation, and through the processes of chondrogenesis, osteogenesis, and eventually remodeling. And periosteum, we've studied in our laboratory, and many others really believe that this is a primary source of cells in the regenerative process. This is evidenced by clinical findings where the periosteum is activated very early in the fracture healing process. And the periosteum as a tissue, although very thin, there's a cambium layer that is adjacent to the bone. And the cambium layer is a very rich repository of mesenchymal stem cells. This is work that Sean O'Driscoll did many years ago. This is an explant of rabbit periosteum where he treated it with TGF-beta. And you can see that essentially all those cells in that cambium layer have the ability and potential to undergo differentiation in the bone-forming tissues. One of the things that's been found is, again, that periosteum in adults is much thinner, and this may account for part of the reason why children heal much more readily than adults. We've utilized a model of periosteal bone transplant to try to understand the process of these cell populations. And in this model, we take a 4mm segment of bone, and we can implant a live autograft that has intact periosteal lining cells. It's very thin in a mouse, and you can't isolate these cells primarily, but they're along the bone surface. Or we can process the graft by freezing it and drying it and reimplanting it. And in that case, it's absent of tissues. This tissue is absent of any cells along the surface. And when we perform these studies with the live graft, what we observe is that at 2 weeks, there's a very robust formation of bone along the surface of these grafts. In contrast, when we take the processed graft, interestingly, we see an absence of a cellular response. So in the absence of the transplantation of the cells, the same autologous bone tissue is not associated with the regeneration of bone, indicating again the importance of these cell populations. To try to define this further, we used our transplantation model, taking advantage of the Rosa 26 mouse, so we could map the stem cell fate of the progenitor population. And this demonstrates this experiment where on day one, again, there's a very thin layer of periosteum. There's proliferation and accumulation of the cell populations. And eventually, you can see by day 10, there's the onset of chondrogenesis and differentiation into bone-forming tissue that will then subsequently drive the reparative process. We observed that these periosteal progenitors are multipotent. They can form cartilage, endothelium, as well as bone cells in the regenerative process. And one of the things that has limited the understanding or ability to study periosteum is difficulty in harvesting the cells. So the model that we've developed to isolate primary periosteal stem cells is to take advantage of this transplantation model, and we expand the cell population in vivo for 5 days, and then we're able to isolate these cells. And this is a case where we used a GFP donor mouse so that we could characterize the cell populations. And this shows an experiment where we use FACS analysis to get out specifically the GFP positive populations. In cell culture, they're green fluorescent cells as expected, but under different conditions, they can undergo differentiation into adipose cells, bone cells, and cartilage, and they respond to those types of signals that are involved in bone regeneration and reparative events. When we also implanted these into nude mice, and as you can see in nude mice, these cells participate and drive reparative processes, and they express these markers of stem cells. Now, clearly, this gets to the question about how does aging or diseases potentially alter these cell populations. In this experiment where we did transplantations and we transplanted young periosteum into young mice or aged periosteum in aged mice, and what we were able to show in this experiment, this is not yet published, but that the aged periosteal cells and transplanted into aged mice have a reduced responsiveness, a reduced capacity for bone regeneration. These mice also, if you look at aged mice, these are 1-year aged mice compared to young, approximately 8-week-old mice. They have a delayed appearance of callus, fracture healing or union across the site is delayed, and remodeling is markedly delayed in these mice. In order to try to understand the cellular processes, we used a tibial osteotomy model where we make an osteotomy, fix it with a pin, and the advantage of this is it gives us very, very elegant histology. And this is an experiment where we looked at the periosteal response at various days at a fixed point from the osteotomy site. And what you can observe is that there is a much more robust response of the periosteum in young mice compared to aged mice. And these cells then can subsequently, after accumulation, begin the differentiation process much earlier. They have the reduced responsiveness to PTH. In this experiment where we took the young mice and we treated with PTH, and PTH activates the stem cell populations along the surface of the bone, and it does it both in young and aged mice, but one of the key features is that you can see that there's a proportionate reduction in the responsiveness of aged mice to PTH. So not only do they have a reduced response to the injury signal itself, but the anabolic agents that can augment the injury response also is a reduced activity. Now, we couldn't do our standard femoral 4mm graft transplantation because, as you know, with aging, there is periosteal bone formation. There's a mismatch in the size. And we tried that, and we got telescoping of the aged bone on the young bone. So in order to look at transplantations from young into aged and aged into young, we took advantage of a unicortical bone transplant model. This was done by Kiminori Yukata as a postdoctoral fellow in my laboratory. And this is work that is, again, being seen for the first time here. This shows the transplant. And when we did these various transplants, one thing to note is that there's both probably cell autonomous and non-cell autonomous effects, so that the young mice are much more supportive of bone formation than is the aged mice. So the young grafts do much better in young mice than they do when they're put into the aged mice. So the aged mouse is a less able host to promote bone formation. What's very interesting, though, is that when we put the aged bone graft into the young mice, the responsiveness was as good as it was in the young mice. And in fact, although we don't have significance here, there's a suggestion that it might even be a little bit increased. So there's probably some effects clearly that are systemic or from the animal that are non-cell autonomous. At the same time, we think there are some cell autonomous effects, and one of the things that we've studied in our laboratory is COX-2 and its regulation in periosteal stem cell populations. Mesenchymal chondral progenitors, as is shown in this in situ hybridization, they make COL2 in the fracture callus. They also have it in the growth plate. What's interesting is that COX-2 is produced in fracture callus, in mesenchymal chondral progenitors, and in the chondrocytes, immature chondrocytes, but not in the growth plate. So again, there are differences between reparative processes in terms of the actual drivers of various signals compared to developmental processes. And this clearly demonstrates that. To understand the role of COX-2 in early fracture repair in these stem cell populations, we used the COX-2 knockout mice and looked at these very early events. And one of the things that we observed is that the knockout mice have a marked reduction in the proliferation of cells at day 3. It's about a 50-fold difference in the number of proliferative cells present, and this persists. In order to define the cell autonomous effect again of COX-2, we used the COX-2 floxed mice, and we crossed this with the COL2A1 Cree and PRX Cree. We obtained the COX-2 from Harvey Hirschman, and we looked at the tissues, but we also looked at the stem cells and their ability to differentiate in cell culture. And COX-2 loss of function in these periosteal progenitors result in reduced fracture healing. This is at day 14. The effect is most profound in the PRX1 Cree. And what you see is that, again, there is less formation of bone. The mesenchymal tissue has delayed accumulation, and subsequently, following that, delayed differentiation into bone tissue. Now, we isolated the periosteal stem cell populations primarily from the PRX1 Cree mice, and we looked at the loss of function of COX-2. And what we observed is that there's a reduction in alkaline phosphatase activity in basal conditions in the knockout mice compared to the wild-type mice. And then if we look at signals that activate the stem cell populations and cause differentiation in the bone, again, the wild-type mice have a much more robust responsiveness compared to the COX-2 floxed periosteal stem cell progenitor. So this clearly shows that there's some cell autonomous effects that are important. And one of the questions is, how does this relate to diseases and aging? And one of the things that we have studied is that COX-2 production is reduced with aging. So this may be one of the cell autonomous effects. And also these can act as paracrine factors. Again, in our transplantation model, adjacent periosteal cells can produce PG2, the metabolite of COX-2, to help drive this process. I want to spend some time on muscle-derived stem cells. Muscle has been known to be very important clinically in fracture healing. This is a paper that looked at a studies that looked at compartment syndrome, where there's a muscle injury in the context of a fracture, and the rate of non-union is markedly increased. Delayed union and non-union is markedly increased if there's been concomitant vascular injury to the muscle. And this also has been shown as well with open fractures having about an 8.2-fold relative risk of a non-union compared to closed fractures of the tibia. So in terms of the various components of the soft tissue envelope, it's evident from experimental studies that muscle is the key component. And this is a paper or study that was done that looked at the relative contribution of either the fascial component associated with an open fracture where there was injury to the periosteum. They injured approximately 3 centimeters of periosteum on either side of the 3 millimeters of periosteum on either side of the fracture site, and they put in this polymer membrane that could block the cell populations from the fracture site, but would let any factors released from those cells have access to the fracture environment. And what was observed is that when the muscle component was present, that you had normal fracture healing, but if you blocked the muscle component, but permitted faster cutaneous tissues to be there, so there was normal revascularization in the area through these tissues, you didn't get healing. So the muscle itself was shown to be an important component in driving this reparative process. And this again shows some of the histology from this, where with the fascial barrier, the muscle component, which is located along the back here, the muscle component is on there. There's no driving of there is repair present here, and then with the muscle barrier, where the muscle is absent, there's no evidence of a repair in this process. Now, the role of muscle in fracture healing can be multifold, and can be muscle-derived stem cells, growth factors, or revascularization. And the concept that's beginning to emerge is that muscle may be particularly important in the setting of injury to periosteum. So it's actually been referred to as a secondary periosteum. Some of the work has been done by David Little in his group, and he performed the following model where he used the MyoD-Cree reporter driving human alkaline phosphatase, which is heat labile, and then closed fractures and open fracture with a periosteal injury, and then looked at the cell fate in these populations. And what this group showed from Australia is that in a standard closed fracture without extensive injury to the periosteum, that the muscle cells were not a major contributor. In fact, they saw very, very little contribution of muscle progenitor cells in the reparative process. However, when they caused injury to the periosteal tissues, they saw that the majority of the healing process was driven, in fact, by these precursors. And in fact, these precursors had the ability from muscle, these MyoD positive cells, to differentiate into both cartilage and bone tissues. What they showed is that there's a decreased contribution of these lineage cells over time. So the secondary remodeling process, the initial process is this accumulation and proliferation of these mesenchymal progenitors. The secondary remodeling process is probably related to periocytes that come in through the vascular supply, and that's what they show as well. They also demonstrated in this control experiment that when they looked at an intermembranous bone formation model with an injury to the cortex in the proximal tibia of the mouse, that there was no contribution of these muscle-derived stem cells to defect healing. Now, again, there are signals that are extremely important in driving this process. And this is a paper that was published in December in PNAS. And they showed, as not surprisingly, that muscle-derived stem cells have markers of stem cell populations, and they can differentiate clearly into bone and cartilage. What's very interesting, though, is they isolated the muscle-derived stem cells, and they did it in the setting of an injury to bone where there was a fracture to the bone. In that case, when they isolated these cells after fracture, the cell populations from muscle expressed alkaline phosphatase and had alizarin red staining, demonstrating evidence of bone nodules. However, when they isolated the same cell populations in a sham operation or in a normal mouse, they didn't get any bone osteoblast differentiation. So this suggested, again, that there's something in the injury process itself, either cell autonomous or non-cell autonomous, that's driving the ability of muscle to respond and drive a regeneration process. They took supernatant from human bone fractures, and in co-cultures with the murine muscle-derived stem cells, they showed, in fact, that these bone fragments from fractured human bone could drive the process. But when they did it in the absence of fracture with control, they weren't able to see this. What they identified through a series of looking at various cytokines is that if they blocked TNF-alpha activity, they would prevent these muscle cells from undergoing differentiation with accumulation of alkaline phosphatase, and that they could do a gain of function in these muscle-derived stem cells with TNF-alpha. So this suggests that TNF-alpha is a signal associated with muscle and bone injury in combination that helps to drive the ability of muscle cells to sense the injury environment and begin to have a response to regenerate the bone tissues. Now, then they finally did a gain of function experiment in the mouse where they injected recombinant TNF-alpha, and they observed, again, that this could enhance the fracture healing process. And they looked at the callus mineralization, and again, the recombinant TNF-alpha had an effect on these stem cell populations in vivo in a setting of fracture. Now, circulation-derived stem cells are another question that's out there, and this is an area that Sandeep Khosla has had a major impact on. And Sandeep has shown that there are circulating stem cells with osteoblastic potential. These are cells, and these cells tend to be higher in younger populations where there's more active bone formation. These stem cells can express bone markers in cell culture. And he has also shown that when they're injected into nude mice, that they can form bone and cartilage tissues. So in fact, they are stem cells that can be involved in reparative processes. In a recent paper, this is currently online, it shows that these circulating osteogenic precursor cells are reduced in diseases, for example, type 2 diabetes. So one of the questions is, how do these contribute to the repair process? And one of the models that has been used to look at this is the peribiosis model. And what this just shows is a control experiment where over the course of about 2 weeks, there is equivalency of the blood between the two partners, and so that there's a mixture and sharing of the peripheral populations of cells after 2 weeks. And the model that was used here was after 3 weeks of peribiosis, a fracture was done so that there would be ability to take a look at these populations. And this was done by George Mushler's group several years ago. And essentially what they showed is that there are GFP positive cells that go to the fracture site, and they can express alkaline phosphatase. However, when they looked at this, the percent of cells is very, very small. So the question is, how well can these cells actually drive that process? And if you take a look at the alkaline phosphatase positive cells in total, a very, very small population of cells actually involved in the bone formation process are from the systemic circulation. So the questions really out there is how do they circulating cells influence repair? And one of the models or one of the concepts come out is that they may actually secrete factors or bring other types of characteristics into the local environment to help drive this. This is a very interesting paper that was in Nature Medicine last month, and it's a little bit off our field, but it's very relevant, I think, to the discussion that we've been having. This group, and what I show is this schematic here is from Darwin Prokop, who wrote an introduction to the article for Nature Medicine, is that this group used bone marrow stem cells to and put them into lung following a lung injury. And they were able to show that actually mesenchymal stem cells could help in the reparative process of lung tissue. But the way it did it is that these mesenchymal cells attached to the the surface of the epithelial cells, and through connexin 43, they actually transported mitochondria into the lung cells. And it was the mitochondria that actually drove the reparative response. So this shows that, again, these stem cell populations can have very interesting effects. And how does this relate, again, to our discussion? And one of the things that has come up again is bioenergetics. And bioenergetics is very, very important in stem cell populations because there is a transition from glycolysis to oxidative phosphorylation that is very necessary for the differentiation process. Finally, I want to just finish by just mentioning vascular endothelium-derived pericytes. This is from a review that, again, this was from Sandeep a few years ago. And what he has shown, and I think what is also the model that's recognized in reparative processes as well, is that these pericytes are probably very important, but more important later in the process. So once there has been accumulation, differentiation of stem cell populations, and you've really initiated and driven a repair process, these cells come in with the vasculature, and they're probably involved in the secondary remodeling processes and final bone formation. So the questions that I came up with are the following. I'm not going to go through these again. We're going to have this so we can get on to some discussion. I want to thank the people in the Center for Musculoskeletal Research that I work with very, very closely. They're a wonderful group and tremendously supportive. These are the various students that are in the center and postdocs doing the work. These are the faculty that I work with in the Department of Orthopaedics and can help drive the clinical aspects. And then finally, I want to acknowledge the NIAMS. They've supported my career from the very beginning and enabled me to have my laboratory program continue over time. So I want to thank the NIH and particularly the NIAMS. Thank you very much.